Yep, that's perfect. So thank you very much for the kind introduction. I would like to start by saying that I am terribly honored to being shortlisted for this award. But I also would like to mention that I don't consider this as recognition of just my work, but also of all those fantastic colleagues and students and technicians that contributed to build up the story that I'm going to tell you. So without much further ado, I would like to start this presentation by telling you what we have discovered over the years about the way plant cells sense oxygen levels and how they cope with the shortage of this molecule. So explaining why oxygen is important is not really a hard task. We all easily realize that oxygen is absolutely essential for our life and for the life of most organisms that populate our planet. We know that oxygen as a terminal electron acceptor is necessary for effective energy production, which in eukaryotic, well, through the possibility of oxidation, which in eukaryotes occurs in mitochondria. Also, the importance of oxygen as a molecule has been highlighted through evolutionary studies that have showed that this being rich in our atmosphere, representing more or less 1/5 of the gas that represents our planet's atmosphere, has pushed life diversity and complexity in terms of different strategies to generate multicellularity. However, despite the fact that oxygen is so abundant on Earth, oftentimes organisms at certain point in their life experience low oxygen conditions that we define as hypoxia. And this is typically caused by limitations in the oxygen availability environment. This is the example, for example, when animals either dive deep in the oceans or climb up in mountains where air is thin. But also in more aerobic settings, extensive metabolic activity might require more oxygen than what is actually available. And this establishes a serious hypoxic condition. Further, it's quite relevant also for human biology and animal biology in general, because tumors and solid tumors are typically associated with hypoxia due to limited supply of oxygen to actively proliferating cells. So very relevant for us to understand how cells sense this molecule. And for plants, this is not much different, to be honest. Also, plants suffer from hypoxia when oxygen supply is insufficient for their oxygen consumption by plant cells. And historically, this has been studied extensively when associated with flooding conditions. This is because oxygen as a gas diffuses 10,000 times slower in water than in air. And this makes so that plants that are submerged, especially when they're not photosynthesizing, then they're restricted in the oxygen that reaches their tissues. And especially for those tissues that do not photosynthesize or photosynthetic tissues under dark conditions or insufficient light conditions are very likely to experience severe hypoxia, if not anoxic conditions. This then generates a serious threat for crop productivity, because most of the crops that we are growing worldwide are actually not able to cope effectively with such conditions. And indeed, depending on the year and meteorological events, flooding can constitute one of the major risks to crop yield worldwide. So this is the reason why we study how we can cope with low oxygen availability and sense when this condition is established. So this is a work that has started actually several years ago, more or less 15 years ago, with my master thesis as a student at the University of Pisa, where I was using Arabidopsis as a model species to study and characterize two transcription factors that belong to the ethylene response factor family. They belong to the subgroup 7, and they are induced by hypoxic conditions. And actually, this is a story of frustration and hard work not leading to much. Because as hard as I was trying to either overexpress, as we see here, or study mutations in these genes, I could never see much of an effect. And I could not see a strong accumulation of the proteins when these were overexpressed. So it turns out that probably I should have focused on other members of the same group of transcription factors that are not induced, but are rather constitutively expressed in the plant cells. And altogether, all the 5 members share very conserved N-termini that contain a cysteine in the second position. And this revealed to be crucial to understand why I could not see much of an effect. So having a cysteine in the second position is typically unfavored for plant proteins and animal proteins. And I wasn't aware of this until I read a review by Emmanuel Graciet, at that time a researcher at Trinity College in Dublin, describing, specifically in plants, a proteolytic pathway that was discovered in 1986 by Andreas Bachmeier and Alexander Krashovsky that is called the N-degron pathway. And essentially it dictates the stability of a protein depending on the N-terminal amino acid that is exposed by protein. And in particular, when proteins contain the cysteine in a second position, these proteins are supposed to be subjected to removal of methionine co-translationally by methionine aminopeptidases, exposing cysteine that in the presence of oxygen can be oxidized, and then this creates a substrate for the activity of arginine transferases that would add an arginine to the cysteine. And the arginine, in turn, is recognized as a destabilizing residue by N-recognin or single subunit E3 ligases that polyubiquitinate the protein and send it for degradation. So this was sort of my eureka moment when I realized that this matched perfectly with my URF7 transcription factors. And this was the first time that an in vivo substrate was hypothesized. Before, all the research was done only with synthetic reporters. So we tested whether indeed preventing oxygen access to cell would stabilize these transcription factors. And here you see one of them called RAP2.12, and we could indeed verify that hypoxia stabilizes this protein and reoxygenation instead favors its degradation. We also could obtain exactly the same effect by knocking out the enzymes that carry out the subsequent modifications that lead to ERF7 degradation. And at the genetic level, if we were looking at genes that are induced under hypoxic conditions, we could see that they are already upregulated, at least to a certain extent, in these mutants that are inactive in the N-degron pathway. I have to say that this discovery was in parallel made from taking it from a completely different perspective by the group led by Mike Holdsworth at the University of Nottingham. So we also identified an additional regulation occurring on the ERF7, and in particular on RAP2.12. We observed that this was able to heterodimerize with membrane or peripheral membrane proteins called the acyl-CoA binding proteins. We verified this interaction both by histohybrid and molecular fluorescence complementation. So this is a bit unusual for a transcription factor to be localized at the plasma membrane, and we would expect it to relocalize into the nucleus at a certain point. Indeed, when we were fusing RAP2.12 with a fluorescent protein that can be photoconverted from green into a red fluorescent protein upon UV light exposure, we could verify what is the contribution of old protein and newly synthesized protein. So before photoconversion, we would have just green labeled RAP2.12, after photoconversion, this would be converted to red. I hope you can see that. The arrow line, the white arrow would indicate the position of the nucleus. So after hypoxia, we could see both red and green fluorescence, indicating that both old and newly synthesized proteins contribute to nuclearly localized ERF7. And this probably is released upon hypoxia. A few years later, we also speculated that the oxidation event on the N-terminal cysteine might be non-spontaneous, as previously suggested, but rather enzymatically catalyzed. We focused on a specific group of proteins at that time without a known function, but which we putatively called plant cysteine oxidase. And we were in particular interested on two of them that are induced by hypoxia, and we suspected that might create a feedback mechanism to repress ERF7 activity. So what we did was expressing this protein in an heterologous system in E. coli, purify them, and verify that they can indeed oxidize cysteine to cysteine sulfenic acid, incorporating two oxygen atoms, but they could also do that better, not just on free cysteines, but rather on peptides containing specifically a cysteine at the N-terminus. And also genetically, we could confirm that, again, inactivation of the genes coding for these would stimulate the expression of anaerobic genes already under aerobic conditions. So these together contributed to define a new enzymatic process that oxidizes cysteine at the N-terminus. So if I summarize all this information together, and I put it also together with other discoveries made by colleagues in the subsequent years, we have a system in plant cells where ERF7 transcription factors are constitutively produced, but also degraded in an oxygen-dependent manner via the endogram pathway. And part of it escapes this degradation by being retained at the plasma membrane through the interaction with peripheral proteins. But upon a hypoxia, the ERF7 enter inside the nucleus, and they activate anaerobic genes until oxygen comes back again, and the protein is degraded. If now we compare this system with the oxygen sensing system that was discovered about 20 years earlier and was awarded the Nobel Prize for Medicine or Physiology to Bill Kellin, Greg Samantha, and Peter Ratcliffe, we see quite some similarities. So in both cases, we have a strategy that relies on degradation of a transcription regulation, and this degradation is oxygen dependent, so that hypoxia stabilizes it. In plants, these are the ERF7 transcription factors that are regulated through the activity of PCOs. In animals, instead, it is a BHLH transcription factor called EF-alpha, which is targeted for degradation by hydroxylation on specific prolyl residues by PHD enzyme. So it seems that this is an effective way to control responses to hypoxia. But the reality is that this is a bit more complicated. And we realized it a few years ago when we collaborated with Peter Ratcliffe and Emily Flashman here in Oxford. And we were looking for potentially enzymes able to carry out the same function in animal cells. And we identified one of them called ADO, or cysteamine dioxygenase. And we confirmed that this enzyme is required to destabilize RGS4 and RGS5. These are G-coupled receptor proteins, regulating proteins, which are stabilized under hypoxia. And this hypoxia stabilization for destabilization requires ADO. So this protein is stable in an ADO knockout mutant. So currently, enzymatic and terminal oxidation of cysteine exists in animal cells, but is not used to control transcriptional responses. So we can then sort of modify our initial hypothesis and imagine that the last common eukaryotic ancestor of plants and animals must have contained both oxygen sensing enzymes. But apparently the divergence between plants and animals had led to the recruitment of either one or the other to control transcription responses. But also, if we look at the different substrates of these enzymes, we see that have there has been a progressive increase in this recruitment of substrates, which nicely correlate with the complexity of organisms in terms of different cell types that constitutes them. So this leads us to speculate that potentially these oxygen sensing mechanisms arose rather to face the need of understanding and coping with endogenous oxygen gradients created by multicellularity, rather than responding to environmental changes in the environment, which is a need also shared by unicellular organisms. And in animals, it is known that stem cell niches are hosted under hypoxic conditions. We found out that also in plants this is the case. So using both naturally occurring promoters and synthetic promoters able to respond to hypoxia, we verified that highly proliferating organs, such as the shoot apical meristem here, the lateral root primordia, or crown galls, which are highly proliferating cells, were also experiencing hypoxia. And we tried to understand why this is the case and if this is integrated into the developmental program. So when we looked at lateral root primordia, we could verify that this hypoxia is established quite late in the development of the primordium. We believe this is associated with cell proliferation, and this can be prevented when we expose primordia to hyperoxy conditions. And cutting a very long story short, we believe that the increase in cell proliferation caused by increasing oxygen in auxin signaling when the primordium needs to be established from the pericycle is leading to an activation of or an establishment of hypoxia and activation of ERF7 activity. This is then translated into the developmental program by the ERF7 inactivating by dimerization the auxin responsive factors, and so terminating the lateral primordium program and facilitating the establishment of the meristem, but also preventing nearby lateral or primed pericycle cells to become lateral root primordium. Finally, we also verified these and the occurrence of chronic hypoxia at the level of the shoot apical meristem, focusing on a shoot apical meristem specific protein, the little zipper protein 2, which we verified is under control of an oxygen-dependent branch of the endegron pathway, so that it can be stabilized by hyperoxia and destabilized by stabilized by hypoxia, destabilized by hyperoxia. And we believe that ZPR2 is there controlled by the endogenous hypoxia being established to control the activity of HD-ZIP3 transcription factors that contribute to regulate the rate of new leaf production. So with this, I would like to conclude and leave you with three take-home messages. The first being that I hope I convinced you that N-terminal cysteine oxidases are conserved sensors of oxygen levels both in plants and in animals. And then oxygen-dependent fertilizers currently is a very successful strategy to regulate transcriptional responses to hypoxia, again, both in plants and in animals. And finally, that in plant tissues, chronic hypoxia can be established as part of the developmental program, and this informs the cells about their fate and their activity. So what comes next then? What I'm doing and what my team is doing at the moment is using the synthetic biology framework and expanding this across the three different kingdoms of eukaryotic life to try to study new mechanisms for oxygen sensing, but also establish molecular devices able to generate desired responses in plant cells, for example, responses to flooding conditions. And finally, I would like to conclude this presentation by thanking everyone that was involved in this. On top here, we have my group. It's impossible to give tribute to everyone involved. I have here by name, so I had here all their pictures, but I would also like to acknowledge the institutions that hosted and supported me throughout this journey. And of course, I will be happy to take your questions.
Okay, thank you, Dr. Likossi, for the very interesting and nice talk, and also perfect time control. You're just right on time. And so now let's move on to our Q&A session between our finalists and the committee members. So everyone in the committee, if you do have questions for Dr. Likossi, please feel free to open up your microphone. Thank you.
Hello, I'm Xiaofeng. It's very nice talk. Really enjoy listen to your talk. I have only one question. Have you ever looked at all these oxygen sensing enzyme or sensor or other proteins? Have you looked at any other species regarding their growing at a high mountain, for example, with low oxygen, to see if there are any evolutionary changes or conserved of this oxygen sensor, especially at the low oxygen condition? See other species of the protein.
It's a very good question. Thank you. So I should say that we are looking at indeed at different species, especially quite far related from the angiosperms we are studying. In particular, we are looking at mosses, ferns, and we are also looking at species, as you were mentioning, that might be adapted to environments that are especially challenging for their oxygen availability. I should not take too much credit on this, but a group of my postwork really recently published a marvelous story in Nature about the role of these ERF7 transcription factors in regulating the adaptation of plants to altitudes. What I think they did not see is differential polymorphisms at the levels of the ERF7 sequences or the PCO enzymes. So there might be other components that also contribute in an accession-specific way or a species-specific way.
Okay, thank you.
Hello.
Hello.
A very nice talk. I enjoy a lot. I just have a short question. You know, in plant macro interaction, the ROS was generated from oxygen used to defend against the pathogen. That means in that tissue, the low nitrogen oxygen will generate. Did you find any, or in other lab, do you find any evidence for the low oxygen sensing system in plant macro interaction?
So I should not take credit for this. I think there are at least two aspects that have been explored so far. The first is in defense, the same direction. And it seems that, for example, that herbivores or necrotrophic pathogens are able or are causing an enhancement of cellular respiration nearby their site of aggression. And this stimulates the stabilization of ERF7 transcription factors that therefore provide a defense. Several of the genes that are activated by ERF7 transcription factors include defense genes. But another aspect of a plant microbe interaction that needs to be explored in terms of oxygen sensing is probably nitrogen fixation. So we know nitrogen fixation requires low oxygen conditions locally in the nodule for nitrogenase activity, but at the same time, oxygen is required for the energy needs of this enzyme first and in general of the plant and microbial cells. So the question that is still open is whether oxygen sensing in plants is activated under the micro-oxy conditions established in the nodule, and whether oxygen sensing participates in this communication between the microorganism and the plant cells.
Okay, thank you.
Thank you.
Hello.
Hi, Christian.
Hi. I just have a question that is maybe a little bit far off, but I was wondering, how are plants dealing metabolically with hypoxia? How does a cell metabolically deal with hypoxia? And is there any evidence for sort of oxygen transport mechanism, not in the sense of a blood transport, but maybe in some sort of concentration release mechanism, something?
So let me address first the part about metabolism. How do plants cope, or plant cells cope with hypoxia? It has been probably one of the earliest goals to be studied, and how essentially low oxygen sensing was approached. And I mean, we know that cells activate fermentation to support glycolysis and regenerate oxidized NAD. The contribution of the two is debatable depending on the species. For example, alcohol dehydrogenase is absolutely necessary in maize. Maybe in Arabidopsis, this is less so. There are other pathways that contribute to that. Hemoglobin seems to be required to generate a nitric oxide cycle that sort of substitutes oxygen for as a terminal oxygen acceptor. And alanine aminotransferases are also typically genes or proteins that are activated under